Grade 10, this is the second lesson in chapter 6. It's chapter 6, section 2 in your book, and we're going to talk about meiosis. Meiosis is the process by which the cells divide in order to have half the number of chromosomes. We already talked about the diploid cells and the haploid cells. Diploid means they have full number of chromosomes, which is actually double the number that is present in your sex cells. While haploid means that they have half the number of chromosomes. Why do they have half the number of chromosomes? Because when the ova meets the sperm, they are going to get to the full number of chromosomes. How do we get to this? We have two types of division happening in the body. We have the mitosis and meiosis. Mitosis is the division that is happening in all of your body cells. Why do we have mitosis in all of the body cells? Because we want all of the body cells to have full the number of chromosomes. So for example, if I'm going to take a sample from your hair, I'm going to get full number of chromosomes. If I get it from your skin, if I get it from the inside of your mouth, wherever I get the sample, we're going to find that they have full number of chromosomes. Meiosis only happens within the sex organs because they lead to the formation of cells with half the number of chromosomes, which are called the gametes, that are going to be used to form a full individual with a full number of chromosomes when this individual is mating with another individual. So how meiosis happens? How does it work? You can think of meiosis as a mitosis happening twice in a row. So mitosis starts with 46 chromosomes in our bodies and ends with 46 chromosomes. In meiosis, we start with 46 chromosomes, but we end with 23 chromosomes. In meiosis, we have two levels. We have two levels, meiosis 1 and meiosis 2. The second division is necessary to maintain the right amount of chromosomes, the right number. In the end of meiosis 1, you're left with two cells that have 46 chromosomes. And then in meiosis 2, which is the second part of the process, the two cells from meiosis 1, they are going to divide again, resulting in 4 new cells. But these 4 new cells, each one will only have 23 chromosomes. So the cells with 46 chromosomes, which were diploid, they are going to form cells with 23 chromosomes, which are haploid, and they have half the number of chromosomes. So here, as we said, we only going to move from 46 chromosomes to half the number, which is 23 chromosomes. Why do we need to do this? Look at this picture. If we keep on having, if our sex cells had the full number of chromosomes, which means if they were diploid, each generation will have double the number of chromosomes. So we need half the number of chromosomes in the sex cells to make sure that each generation keeps the same number of chromosomes. This is one of the main aims of meiosis. Okay, so here in meiosis, this is the first part of meiosis, which is called prophase. In prophase, the nuclear membrane is going to break down. The centromeres and the centrioles, they are going to begin to move. The spindle fibers, they are going to start to assemble. Remember when we said the centromere and the centrioles, they are present within the nucleus. And then the duplicated chromosomes. What are the duplicated chromosomes? Each of the chromosomes is going to form a copy. It is going to duplicate. It's going to form a copy of itself. Okay, the duplicated chromosomes, they are going to condense, which means they are going to collect themselves, and the homologous chromosomes, they begin to pair up. What are the homologous chromosomes? I have 2 chromosomes, for example, for my eye colors. They are going to be standing one beside the other. These are the other chromosomes, for example, responsible for the skin color. So they are going to be collecting and they are going to be condensing. Then we have the metaphase 1. In the metaphase 1, the spindle fibers are going to align. Here, what does this mean? These are the spindle fibers, and in these spindle fibers, they are going to arrange the chromosomes, and the chromosomes are going to be literally hanging from these spindle fibers. So they are going to align the homologous chromosomes along the cell equator, which is the middle of the cell. Each side of the equator has chromosomes from both parents. Then we have the anaphase. The anaphase, the paired chromosomes, the paired homologous chromosomes, they are going to separate from each other and move toward opposite sides of the cell. The sister chromatides, they are going to remain attached. So here they are going to separate and each is going to go to one end of the cell. Now we got to the telophase. In the telophase, the spindle fibers, they are going to disassemble, which means that they are going to separate and the cell undergoes cytokinesis. Then we go to meiosis 2. Remember, in meiosis 2, the starting cell still has full number of chromosomes, because in meiosis 1, the chromosomes duplicated themselves before starting. So the 46 chromosomes became 92. So after the division, they became 46 and 46, still diploid. Now we're going to reach to the point where we need them to be haploid. So the first phase of meiosis 1, it is going to be prophase, but it's prophase 2. In this prophase 2, the centrosomes and the centrioles, they are going to move to opposite sides of the cell, and the spindle fibers, they are going to start to assemble. The same thing that happens in the first prophase. Then in metaphase 2, then in metaphase 2, the spindle fibers, they are going to align the chromosomes along the cell equator. Here they are. In this anaphase, the sister chromatides are going to be pulled from each other and they are going to move to opposite sides of the cell. Here we have two cells. Each one of them, they had full number of chromosomes. Remember, in the prophase, we did not see the chromosomes duplicating themselves. They divided only without duplicating. The duplication happened in prophase 1. Okay? So this means that we started with 46 chromosomes in 2 cells. Now each of these 2 cells is dividing. Okay, the chromosomes inside it are dividing and without duplication. So if we started with 46, we're going to end up with 23. Now we reach the anaphase. So in the anaphase, the sister chromatides are going to be pulled to the ends of the cell, and then in the telophase 2, the nuclear membranes, they are going to form again. So here we started with 2 cells, each with full number of chromosomes, and we ended up with 4 cells, each with half the number of chromosomes, and as we call them, they are haploid cells. This process, when it happens within the sex cells, and it leads to the formation of the gametes, we call it gametogenesis. So gametogenesis, it includes the whole process of meiosis and other changes that produce a mature cells. The final stages of gametogenesis, they differ between the 2 sexes. In the sperm cells, the male gamete is much smaller than the egg. The female gamete, which is the female gamete, the sperm cell's main contribution to the embryo is DNA. Okay, and the sperm develops the tail after the full formation of the cell. The formation of the egg is a complicated process, and we're going to learn about it some more later on during the reproduction chapter. This is for meiosis and the formation of haploid gametes, which lead to the formation of diploid individuals. Thank you very much, grade 10.